chat. If you have any questions during the presentation, please use the Zoom Q&A function at the bottom so that we can find your questions at the end of the presentation. It's always helpful for our speakers to, not talk to a black box. So if everybody would just like to drop in the chat, where you're calling from, what you do, what company you work for, those sorts of things. It always helps our speakers feel welcome. What country, what state. Okay, so we've got Kevin Foley from Rannoch Therapeutics, for example, in Boston. It's local to you guys. Deep Cure in Israel, very cool. Chicago, all right. Some former AbbVie folks there. Ventus in Montreal. Hey Fred from Nurix. Okay, so Bay Area. So we've got folks from all over the place.
Wow.
Thank you all for joining us. I'm sure everybody is very excited to hear your talk about a very famous molecule in this industry. Before we get started, I will just give a quick shout out here. Can everybody see my screen?
Yes.
Okay, thank you. So yeah, before we get started with our guest of honor, I just want to give a shout out and a word of thanks to Porton, our sponsor, for making this session today possible. Porton is a global CDMO that provides chemistry and manufacturing services to pharma companies and biotechs all over the world. They specialize in chemistry and enabling technologies to take compounds from the gram scale in preclinical studies all the way to commercial scale on the ton scale and beyond. They help with potent and highly potent API handling, crystallization, enabling technologies like flow chemistry and continuous manufacturing, and do a lot of GMP manufacturing at scale as well. They are sort of the best kept secret in drug development. They have grown significantly over the last 20 years. And last year, I believe they reached $1 billion in revenue across 700 partners. They have about 1,300 R&D staff as of last year and have worked on over 2,000 projects for biopharma. They specialize in both the early stages and all the way through NDA and beyond, with over 400 projects in early development and several large campaigns at the late stage. Many folks in the US probably recognize them from JSTAR Research. Porton is the parent company of JSTAR. And they have a lot of talented chemists there. So we thank them very much for making this possible. With that, I want to introduce our guest today, Andrew. Andrew is a director of process development in the Pivotal and Commercial Synthetics Group at Amgen in Cambridge, Massachusetts. Prior to joining Amgen, he received his bachelor's from Bowdoin College, conducting undergrad research with Richard Brown. After receiving his PhD from UNC at Chapel Hill with Jeff Johnson, he did an NIH postdoc fellowship with Steve Buchwald at MIT. Welcome, Andrew.
Yeah, thank you for having me, Dennis.
Yeah, thank you so much for joining us. So it's always nice for our audience to get to know our speaker a little bit. Can you tell us a little bit about yourself and how you got to Amgen?
Yeah, so I guess my career in chemistry really started back during my undergrad when I had the opportunity to perform undergraduate research at Bowdoin College. Didn't really know too much about the pharmaceutical industry at the outset, but learned to really love chemistry. And shortly after performing some work in the lab, I decided the next step for me was to apply to graduate school, where I ended up going to UNC Chapel Hill, and developed a love for developing new synthetic methods. I've never worked on any total syntheses. I think in terms of my personality, I really enjoy highly optimizing chemical transformations, which ultimately led me to continue methodology development during my postdoc and pursue a career in process development, which I've absolutely loved. One of the things that I found very surprising about becoming a process chemist is getting excited about every aspect about the process, not just the chemistry. I mean, we're focused on optimizing workups and crystallizations as well, and they can be that work can be just as fulfilling as developing a new chemical reaction, which I think some people may find surprising. I certainly did before I started.
Yep, very cool. And for the chemists in the audience, what would you say is your favorite molecule that you've seen in your career?
Yeah, so I mean, I'm probably a little bit biased here, but I'm going to name an Amgen molecule called AMG176, so you all can Google that. So that is an investigational MCL1 inhibitor. It's a highly complex molecule. It has 7 or 8 stereocenters. It's a macrocycle, and I think the mode of action in inhibiting a protein-protein interaction is also very interesting as well. So I encourage you all to go in and look up the molecule. We do have a couple of papers on certain aspects of the synthesis, and hopefully more will be out soon.
Yeah, absolutely. There was a fantastic ACS talk on the medchem of that a couple years ago, which is great. And actually, that's great timing because next month we're going to have a synthesis article featuring that one. So
awesome.
Stay tuned. We'll probably ask you to make sure that we get all the facts right, but it's definitely a pretty cool molecule. And so yeah, last question before we get started. For the folks who are earlier in their careers in the audience, we always like to ask our experienced guests, if you could go back in time to when you started your career in industry and give yourself one piece of advice, what would it be?
Yeah, so I think when I was in graduate school, I would say I was a pretty serious student and didn't necessarily pause to enjoy what was happening around me, enjoy the people around me. I think I kind of learned my lesson by the time I arrived at my postdoc, and I really developed some valuable and fulfilling relationships with the people that I worked in lab with, and eventually became lifelong friends, becoming groomsmen in my wedding and everything. And I think that if I had focused on developing these relationships more than developing my chemistry and my fume hood, I probably would have been better off with respect to establishing this network of friends who that I was able to achieve during my postdoc. And I guess on the topic of postdocs, I know they're not as common these days, but that was one of the most valuable experiences I've had. Met a lot of really great people, learned some new chemistry, and if you have the opportunity to do a postdoc, I wouldn't necessarily view it as a detour or anything like that. It's really enriching and fulfilling experience that can add to your career and to your personal and professional development.
Yeah, that's fantastic advice. So thank you for sharing. With that, yeah, I'll turn it over to you and let you start your talk.
All right.
Thanks, Andrew.
So are you all able to see my screen?
Yes.
Okay, perfect. Well, first of all, I'd like to thank you, Dennis, for the invite to present to the drug hunter community. Just say it's a privilege to represent the Amgen team here while I discuss our efforts towards developing a commercial manufacturing process for Sotorasib, which is marketed in the US as Lumakras and has the Amgen code of AMG 510. So you'll see these names throughout the course of the presentation. Now, before I jump into the chemistry, I do want to just quickly orient you to where I sit within the organization and within the drug development timeline. So I know many of the callers are probably currently working in medicinal chemistry or have at one point in their career. So you're probably very well familiar with what happens in the early phases of drug development, where chemists are really seeking to make many different molecules in milligram quantities and eventually hundreds of grams as you approach nomination of a candidate. Once nomination occurs at Amgen, it then moves into our process development organization, where we're split into an early phase and a late phase process chemistry groups. Now, the early phase group, they're really responsible for developing the first manufacturing process that can deliver multiple kilogram quantities of the investigational new drug, first to initiate, but also to sustain our phase 1 clinical trials. So generally, these deliveries are on the order of 1 to multiple tens of kilos. Now, as the program progresses, it will then move into the late phase process development group, which is the team that I am a member of. And what we'll do is we'll take a look at the early phase process route and determine whether or not we need to redesign the synthesis, or whether we can double down and highly optimize the pre-existing route so that we can enable deliveries of tens, hundreds, or thousands of kilos of final drug substance in order to progress the program through the later phases of clinical trials and eventually commercial launch. So the team that I'm a part of will take a program from late phase 1 all the way through commercialization. So moving into the LUMACRAS story, I'll just briefly, cover, the target that LUMACRAS is inhibiting and why it is significant. So many of you may be aware of what KRAS is. It's one of the first oncogenes that was identified. The KRAS protein plays a role in the cell proliferation pathway, which controls cell division and cell growth. And depending on its state of phosphorylation, KRAS can either promote or stop this cell proliferation cascade. Now, what was identified several decades ago was that certain tumors possessed mutant versions of KRAS, which was causing uncontrolled cell growth. So these KRAS mutants were stuck in the on position, which was causing uncontrolled cell proliferation and tumor growth. And what LUMACRAS sought to do was undergo a covalent bonding interaction with the mutant to turn it into the off position and thereby ceasing tumor growth. So LUMACRAS is the first approved therapy for the treatment of certain types of non-small cell lung cancer that bear this KRASG12C mutation. Now, in terms of the development timeline, this was a very rapidly moving program. We received FDA approval in May of 2021. This was only 2 years and 9 months after the first subject was dosed in our phase 1 clinical trials. And even more impressive is this was only 4 years after the molecule was first synthesized by our medicinal chemistry team out in Thousand Oaks. Now, if you take a look at the molecular structure of Sotorasib, you'll notice that it has a couple of interesting structural features. One is that it bears this atropisomeric axis, where this the bond rotation about this nitrogen carbon bond connecting to the pyridine is restricted, as well as an acrylamide warhead, which is the portion of the molecule that undergoes the covalent bond with the KRASG12C mutant. As I alluded to earlier, there are a number of different cancers that bear the KRASG12C, as well as other KRAS mutations. Non-small cell lung cancer is the type of tumor that has the highest prevalence of this mutation at about 13%, and colorectal carcinoma is at about 3%. But there are a number of other solid tumors that have a KRASG12C mutation that's present in about 1 to 3%. So this really is what provides a compelling case for developing therapeutics that target KRASG12C, which could benefit a patient population that may not necessarily have a lot of other options. All right, so now we'll jump into the chemistry. So Sotorasib, the synthesis of Sotorasib is achieved from the two raw materials shown at the upper left-hand side of the slide, this amidopyridine 1 and aminopyridine 2. And these materials are progressed through 8-step sequence of chemical transformations in order to access the final drug substance. I'll note that this is a completely chromatography-free synthesis. We purify our intermediates through crystallization processes. Now, due to time constraints, I'm not going to be able to go and dig deep into each and every one of these steps, but I do want to first highlight some work that we had done on the earlier part of the synthesis, which was to convert our raw materials 1 and 2 to our first atropisomerically pure intermediate, which we call M4. And the reason why I want to highlight this chemistry is because the team really did a fantastic job with developing a greener manufacturing process to access M4. And this was work that was recognized by the EPA in 2022 with an EPA Green Chemistry Challenge Award. So I'm not going to dig deep into the chemistry here, but just want to show some of the improvements that we were able to make in developing a new manufacturing process to access first the racemic version RAC4, and then subsequently M4. So the first generation of this process required the use of 100 liters of organic solvent per kilo of RAC4 that was manufactured. Additionally, the solvent that we're using was dichloromethane, which I think we all know is not very desirable. The second generation manufacturing process significantly reduced the amount of organic solvent required by about 97%. So now instead of needing 100 liters of solvent per kg, we only needed 3 liters of solvent. So this is a really impressive improvement. Additionally, reduced the amount of water that was used. We're able to eliminate dichloromethane for acetonitrile, and we're able to cut in half the manufacturing time from about 2 weeks down to 7 days. And one point I do want to drive home is that typically greener processes are also more economic processes. So we're using less solvent, we're using less time in our manufacturing plant, and as a result, the second generation process to access RAC4 costs about 75% less than the first generation process. So now that we have RAC4, we need to access the atropisomerically pure form, M4. And before I get into this, I just want to take a small detour and show this slide that I borrowed from my colleague in medicinal chemistry, Brian Landman, which describes why we're focusing on an atropisomerically pure drug substance. So what the medicinal chemistry team found was when they were examining this early lead molecule shown on the screen here, they were able to separate out the diastereomers, one that possessed the M atropisomer and one that possessed the P atropisomer. And what they found was that the potency of the M atropisomer was about 10-fold higher than that of the P. And this is rationalized by, if we take a look at the protein, there's this pink hydrophobic pocket that is highlighted here, which can undergo an interaction with the isopropyl group when the molecule is in the M configuration. So this increases the binding affinity of the molecule with the target. Now this interaction is not present with the P atropisomer, and this is how we explain why we're targeting the M instead of the P. Sorry, let me go back. Now, one thing I do want to note is that this molecule in particular would not necessarily make a very good drug, and the reason why is because the barrier to rotation about this atropisomer is pretty low. You get a significant amount of racemization, or excuse me, epimerization over the course of about a week. So in Sotorasib itself, you'll notice that this ring here bears additional substitution and is also a pyridine instead of a simple arene. Now, in the early phases of our clinical progression, we were achieving separation of the atropisomers using chirochromatography. Now, chirochromatography is actually a very powerful tool that we can use in early phase programs that enables us to access sometimes difficult to get at chiral molecules such as these atropisomers I'm discussing today, which enables us to access the clinic much more rapidly than we would be able to otherwise. With that being said, relying on chromatography for the long term was not going to be a viable option due to cost, cycle times, as well as the amount of solvent that would be required to run columns on what ultimately would be multi-metric ton quantities per year. So we sought to develop a classical resolution, and I'm not going to talk too much about the story at all, but we were able to identify a very cheap resolving agent DBTA, which resulted in a highly efficient classical resolution, producing the desired atrial isomer in greater than 2,000 to 1 M to P ratio. And one thing that you might notice is that what we're making here is not actually a salt, where there's no ionic bonding occurring. Everything is held together through a hydrogen bonding network. So this is actually a 3-component co-crystal between our solvent 2-methyl-THF, our substrate, as well as our resolving agent. If we take a look at the process greenness comparison between a resolution and chromatography, needless to say, the resolution process is far more efficient. If you're interested in reading more about this classical resolution, we do have a couple of papers that we published late last year, so I encourage you to take a look at those. All right, so now I'm going to dive a little bit deeper into some of the later steps of the Sotorasib manufacturing process. First, focusing on advancement of our first atrial isomerically pure intermediate M4 to this molecule here, 6, that is accessed through a chlorination amination sequence. So for this particular step, the early process generation relied on treating a solution of M4 with POCl3 and Unix base at room temperature to affect the chlorination reaction. So this is a non-isolated intermediate chloro M4. We use this the solution of this species directly in the next step, which where we charge the functionalized pyrazine derivative to form our desired product in 66% yield after crystallization. Now, as this process transitioned into the late phase development group, we wanted to see what we could do to improve upon this yield. And the first thing we need to identify was, well, why is the yield low to begin with? And what we observed was that we were getting large amounts of this pseudo dimer that was forming during this chlorination reaction. And this dimer was present in about 15 to 20%. It was actually 2 different pseudodimeric species that were either N-bound or O-bound. And we attributed our low yields to the formation of these dimers. Now, before we could address this yield issue, we needed to take a closer look at the mechanism in which this pseudodimerization was occurring. So we envisioned that there could be 2 primary pathways. The first being proceeding through this phosphorylated intermediate. So the first step of the reaction is phosphorylation of M4 to produce this phosphorylated M4 intermediate, which could then undergo a pseudodimerization by reacting with another equivalent of starter material. Alternatively, we have this pathway B, which is actually what I showed on the previous screen, where chlorination first occurs prior to this dimerization. So we first want to take a closer look at the reaction profile to determine what's the likely pathway. So this is just a reaction profile by HPLC, where we've got our starting material in green, our product in blue, and the pseudodimer in orange. And what we see is at the very beginning of the reaction, about 10 minutes in, we see a significant amount of our desired reaction has occurred, but we see a spike in the pseudodimerization formation, which does not increase to a significant extent during the course of the reaction, which led us to postulate that the pseudodimers aren't forming from the chloro M4. It's actually forming from the phosphorylated M4. If it was forming from the chloro M4, we would expect to see an additional increase in the pseudodimerization as the reaction proceeds, which is not what we observed. So we had a couple of different options for solving this issue here. First is we could put some effort into increasing the rate to convert our phosphorylated M4 to the chloro M4. This could be done potentially by increasing the reaction temperature or by adding exogenous chloride. Additionally, we could make some process changes where we rely on changing the order of unit operations in order to ensure that there is no M4 present in the presence of the phosphorylated intermediate. So this could be done through changing the order of addition. So what we relied upon was both changing the order of addition and running the reaction at higher temperature, which might initially seem counterintuitive, but because we identified the problem being the slow chlorination step, by heating up this step and speeding up the chlorination, it actually significantly reduced the amount of the dimer that formed. So the combination between reverse addition, where we added M4 into a solution of PlCO3, and heating the reaction to 35 degrees resulted in an efficient chlorination reaction. We're able to suppress the pseudodimer formation down to less than 1%. We then charge in our Bach-piperazine derivative and now get our product in 83% yield after isolation. So these process modifications resulted in a significant yield improvement, and it really relied on understanding how these pseudodimers were forming and what we could do to suppress their formation. All right, so now I'm going to move on to the next step where we take our Bach-piperazine functionalized intermediate, which bears a 2-chloropyridine, and we perform a Suzuki-Mayura cross-coupling reaction to install this fluorophenol aryl group. So the early phase process for the Suzuki-Mayura cross-coupling was quite an efficient reaction. We achieved 85% isolated yield using a potassium trifluoroborate salt as a Suzuki coupling partner, palladium DPPF dichloride, as well as dioxane and water as a solvent. Now, I've highlighted these features of the process in red because these were items that we wanted to address as we moved into the later stages of development. So through high throughput experimentation and our typical manual process development and characterization experiments, we're able to improve this process where we substituted the trifluoroborate with this triiobroxine. And the intent or the reason for doing this is because the trifluoroborate can produce fluoride, which has some material compatibility issues when you begin to manufacture in a manufacturing facility. Namely, the fluoride can etch glass, which is something that we wanted to avoid. We're also able to switch the catalyst, or I should say the ligand on the catalyst, to DPE-FOS from DPPF and reduce the catalyst loading down to 0.6 mol%. And then relied on the use of a greener solvent, 2-methyl-THF instead of dioxane, and we are able to obtain our product in 94% isolated yield. Now, I'm going to focus on a slightly different aspect of this process than I did for the previous step, and that has to do with an interesting observation that we made during the course of development. So the way that the process was being run was we had our 2-chloropyridine, which is compound 6, with base and solvent, and we heated this mixture up to 75 degrees. We then charged the catalyst, and then we performed a slow addition of our Baroxene 8. And the reason why we charge 8 slowly is so that we can avoid a competitive proto-deboronation reaction. Then we monitor for reaction completion. So you may find it odd that we need to add the catalyst in at a high temperature. We certainly found it a little bit odd. And the reason why we did this is because if we add the catalyst up front at the beginning here prior to heat up, or if we charge the catalyst at a lower temperature, we have poor reaction performance. So the reaction will never go to complete conversion if we perform the process this way. So we want to dig a bit deeper and determine why this was the case. So we took a look at how the catalyst or how the pre-catalyst, the palladium DPE-FOS dichloride, was accessing the catalytically active state. So we proposed that it goes through a fairly standard reduction method that occurs in the presence of base and water, which causes reduction from palladium 2 to palladium 0, but also causes oxidation of one of the phosphines on our ligand. Now, what we propose is at low temperature, this ligand can undergo a further oxidation in the presence of trace oxygen and also form palladium black, which is catalytically inactive. And at low temperature, the oxidative addition can't occur. We need to add heat in order for that to occur. So at high temperature, when we charge the catalyst in, the oxidative addition is we propose to be very rapid, and it outcompetes this decomposition to the bisphosphine oxide and palladium black. So we took a closer look at this, and we synthesized this oxidative addition complex, and we did some side-by-side experiments. And in these experiments, what we did was we performed the process where we mixed everything except for the broxine at the beginning at room temperature. So our 2-chloropyridine, our catalyst, and base and solvent were all charged at room temperature, and then we heated to 75 degrees. We slowly charged in our broxine, and then we monitored for reaction conversion. And what we found was our standard pre-catalyst shown by the green line here did not perform very well. It does not reach complete reaction conversion under these reaction conditions. But the oxidative addition complex does. It performs quite well in this reaction, which supports our hypothesis that, one, that this is the catalytically active species, but also that this oxidative addition needs to outcompete a competitive over-oxidation of our ligand in order for the reaction to perform satisfactorily. And we have a lot of supporting details and kinetic studies that I'm not going to touch upon today. So I encourage you to take a look at a paper that we published earlier this year in OPR&D. All right, so now moving on to the Bach deprotection process, where we remove the Bach group from the piperazine portion of the molecule to reveal the free amine. Now, the Bach deprotection is a fairly straightforward reaction. I'm not going to go into any detail on this chemistry, but we'll rather focus on a different aspect of process development, which is the isolation process. So after the Bach deprotection reaction, which is run using trifluoroacetic acid and dichloromethane, we have a solution of a TFA salt of our product. That solution is then added into a solution of an aqueous solution of potassium carbonate with methanol. So this is a reverse quench, which also causes concurrent crystallization to access our free base product. Now, here's a microscope image of the crystals that we obtained from this process. You can see they're extremely small. And as a result, it takes a week to filter on a 90 kilo scale. So this is going to be highly problematic for us because we probably need to run around 15 batches of this step per year on multi-100 kilo scale. And if we need to take a week or more to just perform the filtration, it takes up a substantial amount of our manufacturing capacity per year. So this was a high priority item that we needed to solve. Additionally, what we found was methanol was causing the formation of an impurity. So we could undergo a methanolysis reaction where we lose the entire pyrazine piece and it gets substituted with this methanol. So this was also another problem that we needed to address. So one of the first parameters that we focused on was altering the co-solvent that is used for the crystallization. And I'm reducing a lot of work down just into a couple of slides, but ultimately what we found was that NMP was a superior co-solvent for the crystallization. And I have a few in situ microscope pictures of the crystallization process itself, which I find to be pretty interesting. So immediately after charging in our reaction mixture into the quench solution of potassium carbonate with the NMP, we have all of these droplets of product-rich dichloromethane, which is kind of in a sea of our quench. And over time, what we see is the growth of crystals coming off the surface of these droplets as the quench occurs. And later on, as we continue to age the quench, you can see lots of crystals forming on the surface of the droplets. Now, these are like massive crystals compared to the methanol process. As you can see in the side-by-side comparison, at the same magnification, you can see the crystals that are forming through this NMP process dwarf those of the methanol process. Another interesting aspect of this crystallization is that the addition rate during this reverse quench and the agitation rate has an impact on how large the crystals grow. So if we look at the left-hand side of the graph here, when we have a slow rate of addition in this reverse quench and a slow rate of mixing, we generate these really small, kind of fine hair-like crystals. And as a result, the filtration is extremely slow. You can imagine, hair on a drain just overlapping with each other and just restricting flow of the mother liquor through that cake. And this just is exacerbated as you begin to scale up. And what we found was if we agitate the crystallization faster, as well as charge the reaction mixture during a reverse quench faster, then we get very large, well-filtering crystals. And we're able to run some small-scale experiments and model how long it was going to take to filter on kilo lab scale in order to provide some guidance that we could then validate prior to scaling up. So these results are actually a little bit counterintuitive. Typically, you would expect a more controlled crystallization to provide better crystal morphology. So usually you would think a more controlled addition or potentially lower mixing would provide better results, but that was not the case. And we're still working on our hypothesis for why that is, but we think it has to do with the fact that we are forming these small droplets, and the crystallization can occur in a very uniform fashion if the reverse quench occurs rapidly and if the agitation is very high. So in the final process, we were able to perform the Bach deprotection and then isolation and obtain our product in 95% isolated yield. We improved the filtration time considerably. We reduced down from 1 week on 90 kilo scale to 9 hours on multi-100 kilo scale. So this was a really important improvement that the team made on this process. And hopefully this highlights some of the other things that process chemists work on beyond just solving issues with chemical reactivity. All right, finally, I'm going to move on to the last step of the synthesis, which is formation of the acrylamide by treating our free Bach pyrazine with acrylyl chloride. So on paper, this seems like a very simple transformation. We dissolve up our starting material in NMP, we charge acrylyl chloride, and we get our product. But by design, AMG 510 is a reactive molecule. We want it to undergo a reaction with the cysteine that's present on the KRASG12C mutant. And because of that, we have some issues with overreactivity during the process to synthesize AMG 510. And what we found was our starting material was undergoing a Michael addition reaction with our product to form this pseudodimeric species shown on the right-hand side of your screen here. So we asked ourselves, what can we do to attenuate the reactivity of our starting material so that it doesn't undergo this Michael addition, but will still undergo the desired reactivity with acrylyl chloride. And this led us to start looking at acid additives into the reaction. Now, this isn't an exhaustive list of what we looked at, but just a couple of telling results. So on the left-hand side is the reaction with no acid. Reaction conversion is shown in blue, and the impurity formation is shown in red. If we add an HCl, we start suppressing our reaction conversion to a significant extent once we are up around 1 equivalent. Half equivalent was not effective at reducing the impurity formation, although reactivity was still maintained for the desired reaction. What was kind of the sweet spot for us was the use of TFA. So moving over to one equivalent of TFA, we maintain the desired reactivity while also completely suppressing formation of the pseudodimer. So we're excited to see that. So this process provides the final drug substance in a 95% yield. Now, one thing that we did observe as a side effect of using TFA was the formation of a new impurity. So this trifluoroacetamide impurity, TFA9, shown on the screen here, which is present in very low level of about 0.05%. But we want to assess, how big of a risk is this impurity? Can we maintain it at 0.05%, or was there a risk of there being elevated levels in the future? So we ran some additional, so let me take a step back. So we had a couple of hypotheses as to how this impurity was forming. One was potentially our TFA could be contaminated with trifluoroacetic anhydride, which could be transferring the trifluoroacetyl group, or our TFA could be undergoing a reaction with the acrylyl chloride itself to form a mixed anhydride, which would then transfer trifluoroacetyl group. So in order to kind of assess how this impurity was forming, we ran a series of experiments. Baseline, as shown at the top here, we get about 0.05% of the impurity. TFAA or trifluoroacetic anhydride had no impact on the level of this impurity, which suggested to us this is not proceeding through a TFAA pathway. Varying the reaction temperature had very little impact on the amount of the impurity. But what did have an impact was when we pre-mixed TFA with acrylyl chloride, we see about 0.5% of this impurity. And what that suggests to us is that the reaction is proceeding through a mixed anhydride pathway. So because we were not actually pre-mixing TFA with acrylyl chloride, and under various processing conditions, we never saw the impurity increase greater than 0.05%. We considered this a low risk. But I did just want to show this as an example of seemingly small changes to a process can have unintended consequences in the future. So it's really important to dig deep and take a look at the reaction profiles and the impurities that you're forming as you make process modifications. All right, so I think with that brings me to the end of my talk here. So I had a chance to touch upon all the steps of the process to manufacture Sotorasib, some in more detail than others. If you are curious about learning more, we've published a series of papers over the past 9 months or so. So I've provided the references here, so I encourage you to please take a look. And with that just leads me to thank the team who supported the program over the years. So I want to acknowledge everyone from the process chemistry and engineering team across early phase and late phase development, as well as my colleagues in the analytical groups who really made it possible for us to move this program forward as quickly as we did. And this is just a picture from last year when we received the EPA Green Chemistry Challenge Award, which again we're really excited about. And with that, Dennis, I just want to thank you again for the invite to present to the drug hunter community, and thank you all for joining in the discussion today.
All right, thank you so much, Andrew. If we were live, there'd be a big round of applause from the 130 folks in the audience. Really appreciate you sharing this with us. As a chemist, I saw a lot of mind-blowing things there. I'm going to love, presenting or telling people about the solvent choice and how, of course, NMP was chosen because crystals form on the droplet interfaces, which is pretty cool. So we have some questions from the audience. I'll start with those before I get to some of my own. First, from Colin Morris is, did you look into anything like high shear wet milling and the particles generated from the reverse addition crystallization?
Yeah, that's a good question. That is something that we did consider implementing. I think that, for us, the simpler the process, the better. And we never reached the stage of needing to rely on high shear wet milling in order to improve that crystallization process. But it's certainly a valid approach, and it's one that we've actually taken for other programs that had a really high dependence on mixing. So it's a good question, definitely a valid approach, and I think it would have worked in this case. We just didn't get there.
Makes sense. Next question comes from Fred Cohen. He asks, TFA is pretty expensive. Why not HCl for Bach removal? And I'm sure a lot of med chemists had the same question.
Yeah, so the reason why we didn't use HCl, and it's not something I went into in any extent here, is that HCl has an undesired consequence in promoting hydrolysis of our piperazine piece. So if you use too strong of an acid, then it results in hydrolysis. So of course, yeah, HCl certainly could have been used if it were not for that issue.
Makes sense. Asha asks, have you tried BOP activation for piperazine insertion instead of chlorination?
Oh, geez, that's a good question. I'm trying to think back on what we looked at for that activation. I don't believe, or I can say for certain that the late phase group did not look at using BOP activation. It potentially could have been looked at in an earlier stage that I'm not aware of, and apologies for not having a better answer to that question. I think it certainly could be a valid approach towards forming that bond, but it's just not one that we looked at.
Makes sense. No worries. And a related question is, how did you remove NMP completely at the final stage?
Yeah, so so I kind of left off the last step of the process, which actually is a final form setting recrystallization step. So there's actually some really interesting aspects of that final crystallization process that I did not discuss today, but that is how we end up removing the NMP. What I'll say is that the recrystallization was going to be performed regardless of whether or not we needed to remove NMP, and it was not due to impurity upgrade or anything like that. It was really more geared towards ensuring that we have the correct crystal form of the final drug, but it had the added benefit of also removing the NMP.
Cool. All right, and next question from Delaney Fernando is, also in the Bach removal, you still use DCM. Did you try other solvents for that, or was there a special reason?
Yeah, we did try other solvents, and we would have loved to get away from DCM, believe me. So that the Bach protection, or what I should say is the crystallization itself is a finely tuned process. So if we deviated from, or what we found was if we deviated from the specific solvents that we're using, then we end up back kind of where we started with forming small particles that did not filter well. And ideally, we would have identified something that did not rely on the use of DCM. But I will say that we did not have a lot of time to develop our process either. So this, I think all of what I described to you today was developed over the course of about 6 months, which is a very short period of time for developing a commercial manufacturing process. There are definitely some things that I think we wish we could have done better, that I think we could have done better if we had some more time.
Yeah, makes sense. Bensa asks, might have to go to the slides for this. He asks, have you tried a 2-step process in step 7 with chloropropanoyl chloride addition and dehydrochlorination? Not sure.
So I'm familiar with this approach, and it's an approach that is utilized for Tagrisso, which is an oncology asset from AstraZeneca, where basically you use 3-chloropropanyl chloride for the reaction, and then you treat with the base for the elimination. I guess my argument is that a 1-step process is going to be simpler than a 2-step process. Of course, there were some challenges that we needed to overcome by approaching the 1-step process. And also would like to add that 3-chloropropanyl chloride, as well as the hydrolyzed version, so the 3-chloropropionic acid, they're mutagenic. So using a mutagenic reagent at such a late stage in a synthesis also is not, well, I think it's something that we could have certainly managed. If we were able to avoid it, then that is the route that we'd prefer to take.
Makes sense. I hope you don't mind taking more questions. It's a very popular talk, and there are some very interesting things here. Next question comes from Pingyu Yang, asking, regarding the epimerization study, you have a T-half of 8 days. Was that stability from the solid state or solution?
So I would have to refer back to the MedChem team on where they got that number from. What I can tell you, at least for Sotorasib itself, we did some experimental studies as well as some computational studies to determine the barrier to rotation for Sotorasib. And the way that we did that was in the solution phase. We actually did pursue solid phase as well, taking a small amount of material, actually putting it on a DSC and heating it up to 300 plus degrees to see what would happen. And in those instances in solid state, we actually typically saw decomposition. But in the solution phase, we have a publication on the racemization of P4, which is the waste waste byproduct from the classical resolution. We're actually able to racemize it back into the RAC4 and then resubject it to the resolution process. When we try to determine the barriers to rotation, at least the approach that I would take would be run this in solution phase and collect HPLC data to monitor the racemization over time, and then you can use the I-ring equation to determine what the barrier to rotation is.
Cool, thank you. Dennis Koester has a question that I have as well, which is, can you explain to a medicinal chemist why process chemists like 2-methyl-THF so much? And related to that, I'm curious about the crazy 3-component crystal that you have with the 2-methyl-THF. Is the 2-methyl-THF there, does it co-crystallize in one isomer, or?
Yeah, that's a good question. And it was one that I was curious about at first as well. I think it would be really cool if we're also resolving the 2-methyl-THF, but unfortunately not. It is a mixture of enantiomers within that co-crystal. And actually, we were recently contacted by a company that manufactures an anti-pure 2-methyl-THF. So one of the things I think would be interesting to look at is seeing if there is any impact on the wrong enantiomer of solvent to see if that actually causes a decrease. But it seems like it probably would not. In terms of why we like 2-methyl-THF, well, I can tell you why I like it. I like it more than THF because it can reliably phase split after a reaction when you need to do a workup. THF will not always necessarily reliably phase split. It's also, from a process greenness perspective, it's a more sustainable solvent. It's derived from, I think, lignin, so it's derived from furfural. And it's just exhaustively reduced down to 2-methyl-THF. So yeah, that's the reason why we like it.
Cool, makes sense. All right, a couple more questions. So let me try and pick out a couple more. Let's see here. All right. So Mike Damaso asks, do you see any acryloyl chloride polymers in the last step, and how do you control those? I'm sure a lot of folks working in covalent chemistry have that question as well.
Yeah, so that's a good question. We have not directly observed any polymerization. I don't want to say that it's not happening. It certainly could be, but the way that we semi-quantitatively are able to monitor this through the presence of acrylic acid after the quench. So we are able to observe a significant amount of acrylic acid after the quench step, where we're hydrolyzing that acrylyl chloride. But yeah, I mean, I'm sure if we dug deep, there's probably some polymeric material in there.
Makes sense. And I'll pick one last question from the chat, which is, hi, Andrew. Rick Arts from Arrowhead. KRAS is not unique to non-small cell lung cancer. Are there any other indications on the horizon for 510?
Yeah, hey Rick. So Rick and I worked together on some of this back in the day. So good to hear from you. So yeah, Rick, that's a great question. So right now, AMP510 is being investigated for some other solid tumor types. One that I highlighted on one of my earlier slides, colorectal carcinoma. So I think I'm not sure what stage of clinical trials we're in, but we are investigating for colorectal carcinoma. I know in phase 1, we looked at, I think, 14 different tumor types, just to highlight the prevalence of these mutations in a variety of different solid tumors. So, I mean,